My great privilege to present this year's Translational Year in Review. We've seen tremendous progress despite the challenges of the past year, and I hope to highlight some of these advances and opportunities for further study. These are my disclosures, and I'll begin with several acknowledgements. I want to thank colleagues near and far for the outstanding basic, translational, and clinical research that's being done, and to point out how discoveries across each of these arenas is really fueling the others towards a common goal of improving breast cancer patient outcomes. I won't be able to cover all of the important research in this brief review and apologize in advance for omissions. I want to thank the patients for their altruism and for inspiring this research. We simply wouldn't be able to do translational science without their participation in clinical trials, and it's exciting to see the pace at which correlative science is advancing the field. I'd like to thank the San Antonio Planning Committee for an excellent meeting, as well as my colleagues for critical review of the slides. So I'm going to begin by reviewing advances in hormone receptor positive breast cancer, given that this is the largest subgroup of disease, accounting for upwards of 80% of all breast cancers. I'll speak to resistance mechanisms, anti-tumor immunity, and biomarkers. So we know that the treatment of ER-positive breast cancer comprises interventions that suppress estrogen production and or target ER directly. And while endocrine therapy has considerably reduced recurrence and mortality from breast cancer, de novo and acquired resistance remain common. Increasingly, we're understanding the mechanisms of resistance to endocrine therapy, including somatic alterations in HER2, FGFR, and other RTKs, but also epigenetic changes. In addition to endocrine therapy, we of course have CDK4/6 inhibitors, which are now being moved to earlier stage disease. But again, resistance is common, both intrinsic and acquired, including mechanisms of FGFR1 alterations, AKT, and other downstream targets. And so really elucidating the molecular features of sensitivity and resistance to CDK4/6 inhibitors may ultimately lead to the identification of predictive biomarkers and new therapeutic strategies. And this is being accomplished through both laboratory and clinical-based approaches. So I want to kind of go back to the beginning, because we all know that ER-alpha has been studied for its role as a transcription factor for decades. However, recent work from Zhu and colleagues highlight an unexpected role for ER-alpha as a non-canonical RNA binding protein that in fact can be uncoupled from its DNA binding activity. And so using ClipSeq and functional CRISPR screens, they show that ER-alpha associated mRNAs can modulate the post-transcriptional expression of stress response genes. They are implicated in RNA metabolism, translational control via MCL1, as well as XBP1 splicing. And so together they can bypass anti-tumor mechanisms, really facilitating survival upon stress conditions. And as a result, ER-alpha binding proteins have been implicated in tumor growth, tamoxifen resistance. And so, of course, this begs the broader question, is ER-alpha RNA binding proteins implicated in a broader RNA biology, or do they have a role in early tumorigenesis? And so this will be the course of further study. Now we know that the acquisition of ligand-independent ESR1 mutations in the context of endocrine therapy is a common mechanism of resistance. And genomic analyses have identified both point mutations as well as ESR1 translocations that encode chimeric proteins, whereby the ESR1 ligand binding domain can be replaced by C-terminal sequences from many other gene partners. Moreover, ESR1 mutations can express a unique transcriptional profile that may promote tumor progression. And so in recent work from Ellis and colleagues, they sought to identify gene expression signatures that might discriminate functional ESR1 mutations. They used a combination of in vitro and correlative studies and identified 24 genes with such a pattern. Moreover, they demonstrated that this gene set could predict functional ESR1 mutations in an independent cohort, suggesting that it may have utility for patient stratification. We know that patients with ESR1 mutations may derive clinical benefit from fulvestrant and CDK4/6 targeted therapies together, but ultimately we may need to turn to the development of more potent SERDs to overcome this resistance. And of course, there's much activity investigating these new classes of SERDs currently underway. So FGFR1 has also been associated with intrinsic and acquired endocrine resistance through transcriptional regulation of ER responsive genes in a ligand-independent manner. And sequencing of paired pre- and post-treatment tumors following endocrine therapy has revealed an enrichment for FGFR1 alterations following therapy. Because we can target FGFR, there's been a quest to define biomarkers for FGFR-altered tumors. And in this recent study, the investigators sought to evaluate the association between FGFR copy number alterations and expression, as shown over here in a cohort of patient-derived xenograft models looking at response to the selective FGFR inhibitor rogatinib. And they found that high FGFR 1 through 4 mRNA, but not cRNA, was associated with response. In fact, there's relatively limited correlation at the copy number and expression level. So this led them to develop a composite score that they argue could potentially be used to refine patient stratification. Now, in another branch of this work, the investigators also sought to elucidate the relative clinical benefits of multi-targeting tyrosine kinase inhibitors such as ositinib, and they show that the clinical benefit is largely mediated by the potent anti-angiogenic effect of these drugs. So indeed, while there's a higher response rate for MTKIs versus selective FGFRIs, there is toxicity associated with this, making it harder to move forward in the clinical setting. And so these data suggest that new strategies are needed to target FGFR. And this is especially the case given the relatively modest results for the triple combination of FGFRI with palbo and fulvestrant in metastatic disease. Now, in other work, Arteaga and colleagues have identified the role of nuclear FGFR1 in resistance, again, via transcriptional deregulation. They show that FGFR1 can localize to the nucleus in endocrine-resistant primary tumors, and in vitro and in vivo models, nuclear FGFR1 promotes anti-estrogen resistance. Furthermore, they show via ChIP-seq assays and mass spec that, in fact, FOXA1, the pioneer factor, recruits FGFR1 to transcriptional start sites. And importantly, they demonstrate that while FGF2 enhances the activity of nuclear FGFR1, it is relatively unaffected by FGFR inhibitors such as erdafitinib, and this may explain the relatively limited activity of TKIs against FGFR. So this suggests that perhaps we need new pharmacologic approaches altogether to target the nuclear activity of FGFR1. CDK4/6 inhibitors are, of course, designed to induce potent cell cycle arrest. However, recent studies have also suggested that these agents can exert other effects influencing cancer cell immunogenicity, apoptotic response, and differentiation. And in this study by Watt and colleagues, they demonstrate that CDK4/6 inhibition induces chromatin remodeling and promotes luminal differentiation. Furthermore, they show that this results in the activation of enhancers that overlie long terminal repeats, a class of endogenous retroviral elements, and these are located proximal to interferon genes. So this suggests that ERVs may be implicated in collateral damage activating viral sensing, innate immunity, and this really may further explain the anti-tumor immunity associated with CDK4/6 inhibitors. Now, they further show that CDK4/6 inhibition induces RB-dependent upregulation of AP1 expression and AP1 target genes, as shown over here in Neopilana, but also BCL2L1, BCL-XL, and MCL1. And importantly, BCL-XL inhibitors can reverse the anti-apoptotic state that is induced. And so these data beg the question, should we be contemplating combinatorial therapies such as the combination of CDK4/6 inhibitors with BCL-XL inhibitors and/or immunotherapy? And following on from this theme, Schiff and colleagues have reported an inverse association between response to CDK4/6 inhibitors and interferon signaling pathway activation. And they observe that interferon-related genes are associated with resistance to palbo in cell lines, as well as in the neoadjuvant studies such as Neopilana and NeoMonarch. Furthermore, they show that interferon response genes are associated with poor prognosis in early stage ER-positive HER2-negative breast cancer, as shown in the Metabrit cohort. and that the signature is associated with the activation of immune checkpoints such as PD-1, PD-L1, and CTLA-4, highlighting the complex interplay between CDK4/6 inhibition and anti-tumor immunity. There are currently no established biomarkers to predict response of patients receiving combination endocrine and CDK4/6 inhibitors. And so a key question is whether ctDNA has utility for this purpose. And so in a retrospective analysis of PALOMA-3, O'Leary and colleagues sought to investigate this by studying pre-treatment ctDNA in 459 patients. They demonstrate that high ctDNA, in excess of 10%, was associated with worse progression-free survival in this high-risk population in both treatment arms, namely the palbo and the placebo arm. Now, they go on to show that poor prognostic factors include not only ctDNA levels, but also TP53 mutations and FGFR1 amplifications, which were collectively present in over 40% of cases. And while no alterations were predictive of treatment response, they did demonstrate some associations in particular genomic features with PFS, including the enrichment of CCNE1 gain, CDK4 gain that were found in the palbo treated cases. Now, these data speak to the need for more intensive surveillance, but also escalation of therapy in these high-risk populations. But I will point out that in follow-on work from some of these authors, they furthermore showed that biomarkers of early progression also predict shorter PFS on triplet-based therapies. And importantly, CCNE1 mRNA levels emerged as being associated with resistance in PALOMA-3. So clearly, new approaches are needed to tackle these high-risk cases, and this is an area of much ongoing work. So ER-positive, HER2-negative disease is clearly very heterogeneous. And as of yet, we have a relatively limited understanding of the prognostic and predictive value of the intrinsic subtypes in advanced disease. And so in this recent study from Pratt and colleagues, they sought to perform a retrospective PAM50 analysis of the Mona Lisa phase 3 clinical trials. And they demonstrate that PFS benefits were observed for the ribociclib plus endocrine therapy arm versus placebo across all of the intrinsic subgroups with the exception of basal, which is a relatively small subset. They furthermore show that the HER2 enriched subgroup derives the highest absolute benefit from ribo. And so these data really speak to the power of correlative studies, and these important findings are being further validated. So turning to lobular breast cancer, which is a common but special histologic subgroup of disease, we know that most ILC tumors express estrogen receptor, making them good candidates for endocrine therapy. However, they often become resistant and metastasize to other sites and really are relatively understudied at both the somatic and thermaline level. So in this recent work from Sotnick and colleagues, they hypothesized that ILC-specific ER co-regulators might mediate anti-endocrine resistance, and they studied this through a combination of in vitro assays, including profiling ER-associated proteins by mass spec. This led to the identification of MDC1, a DNA damage checkpoint, as being involved in this process. And they further showed that knockdown of MDC1 suppresses ILC proliferation as well as endocrine resistance. Moreover, they demonstrate that MDC knockdown results in broad ER transcriptome deregulation and really suggests an oncogenic role for MDC1 as an ER co-regulator that's specific to ILC versus IDC, where it may behave as a tumor suppressor. Moreover, they show that DNA damage induces MDC1 and gamma H2X foci formation, and this was sustained in ILC cells. And so this begs the question, might MDC1 be a new target that could aid our efforts to overcome endocrine resistance? And could there be a role for DNA damage response inhibitors in therapy for this subgroup? Now, as ILC is relatively understudied, there are open questions regarding the contribution of pathogenic variants in hereditary gene panel testing. And while it's known that CDH1 variants are associated with ILC as well as gastric cancer, the magnitude of risk has been found to vary between studies due to the small number of carriers. So in this study, Yadav and colleagues sought to systematically investigate this in both a clinical cohort and a population-based cohort. They find that 6.5% of the clinical cohort harbor pathogenic variants, whereas 5.2% of the population-based cohort. And in a case control analysis, they identify CDH1 and BRCA2 as conferring high risk of ILC, whereas CHEK2, ATM, and PALB2 variants were associated with moderate risk. BRCA1 was underrepresented here, and in contrast, there was a tenfold increase in the risk of CDH1 pathogenic variants, prevalence of CDH1 pathogenic variants in ILC. So given the similar frequencies of pathogenic variants, this really suggests that histology should not be influencing genetic testing decisions, and that this is something that should be considered for patients with ILC as well as IDC. So the HER2 amplification defines a distinct subgroup of breast cancer for which considerable translational, epidemiologic, prognostic, and predictive data have been studied. We also know that HER2 can be heterogeneous, and that there's a number of emerging biomarkers on the horizon, as well as an important subgrouping of HER2 low cases. And so speaking to the heterogeneity, HER2 amplification heterogeneity has been associated with resistance to TDM1. And in a study by Metzger and colleagues, they sought to prospectively evaluate how HER2 heterogeneity impacts response to the combination of TDM1 and pertuzumab in the neoadjuvant setting. They surveyed biopsies from 2 treatment biopsies looking at FISH-based HER2 scores, and they found that there were PCR rates of 55% in patients who were not heterogeneous for HER2 versus 0% in the heterogeneous group. And this was after adjusting for hormone receptor status, which is of course important with respect to PCR. Furthermore, they show via single cell ERB2 FISH that the fraction of non-amplified cells is predictive of resistance. And so really these data speak to the potential value of integrating heterogeneity in treatment stratification approaches, and whether we can identify a subgroup of patients who will respond sufficiently well to HER2 targeted therapies alone and therefore can be spared chemotherapy. So we've been interested in the question of HER2 heterogeneity at the proteomic level and sought to examine this by performing digital spatial profiling using a 40-plex marker panel of paired pre, on, and post-treatment biopsies from the TRIO USBO7 study of neoadjuvant trastuzumab and or lapatinib. This was work done with Sarah Hurwitz, and importantly, looking at the pre versus on treatment changes, we see pretty dramatic alteration in HER2 signaling being downregulated and immune markers being upregulated in the PCR cases, not seen in the non-PCR cases. However, at the time of surgery, we do see compensatory signaling taking place, as well as an influx of immune genes. Now, with these data, we were able to establish that on-treatment CD45 measured via DSP or immunohistochemistry predicts PCR with high accuracy and high positive predictive value. And this begs the question, could this on-treatment biomarker potentially be used to inform therapy de-escalation? Of course, this needs to be evaluated prospectively, but these data also speak to the importance of an on-treatment biopsy to understand dynamic changes in tumor and immune composition. Of course, there are numerous other efforts attempting to de-escalate therapy via omission of chemo. And this is being investigated in early stage HER2 positive disease. These include the ADAPT trial, as well as the FERGAIN trial, which seeks to use a PET-based biomarker. So it's really exciting to think about these studies as we seek to find ways to dispense with chemotherapy and to design new ways to reduce the toxicity of such approaches. And so stay tuned in this really exciting area. Now, we know that HER2 can be heterogeneous, and recently a definition of HER2 low tumors that have low expression and lack detectable ERB2 amplification have been come into the limelight. And of course, the question remains, how do we best define these groups? That's of course dependent on the testing approach. We also want to better understand the biology of these HER2 low tumors, which are not addicted to HER2, But what's really exciting about this is that novel anti-HER2 compounds, such as antibody-drug conjugates, including TDXD, are showing promising activity in HER2-low tumors. And so from a therapeutic perspective, the benefit in the setting may well be achieved via alternate pharmacologic mechanisms, including the delivery of targeted cytotoxic agents. And so beyond the ADCs, there's also potentially a role for bispecific antibodies, monoclonal antibodies, in targeting this subgroup that previously wasn't thought suitable for HER2 targeted agents. So this is clearly a very exciting area with much ongoing work. So turning our attention to triple negative breast cancer, one of the hallmarks, the high degree of tumor heterogeneity, and we'll touch on this as well as the importance of understanding the immune microenvironment to develop predictive biomarkers, both for neoadjuvant chemotherapy, but also immunotherapy. And so in a recent study from Manussi and colleagues, they sought to investigate heterogeneity, profiling 8 tumors as well as 4 cell lines using single cell and single molecule DNA-based sequencing. And using this approach, they uncovered many subclones. They found that TP53 mutations were prevalent, often accompanied by loss of heterozygosity and genome doubling. There was transient genome instability and ongoing evolution. And really, they conclude that this has led to a reservoir of subclonal diversity with no single clone being dominant. And this is in line with studies in other tumors that suggest that the subclonal diversity may occur early and represent a potent substrate for evolution in the context of treatment selective pressure. Now, as immune checkpoint blockade has shown modest survival benefits in triple negative breast cancer with only a subset of patients responding, a key question is whether markers in the immune microenvironment may be predictive of response. And so in this study, Hammerl and colleagues sought to investigate the spatial contexture that may mediate this, and they demonstrate that lymphocytes, but not myeloid cells, are prognostic in primary TNBC, and they derive essentially a gene expression signature based on bulk data combined with patients where CD8 positive T cells were profiled. So they're able to really map from excluded, ignored, and inflamed into a gene expression profile, looking at many patients in early stage disease. Now, furthermore, they turned to the TONIC trial where metastatic TNBC patients received anti-PD-1 therapy, and they showed that there is also an association with these spatial phenotypes in TONIC. Notably, there was a relatively small number of responders. They further seek to investigate whether these distinct immune archetypes of excluded, ignored, and inflamed might reflect different paths to immune evasion and nominate different therapeutic strategies, such as the combination of TGF-beta or VEGF inhibitors with immune checkpoint blockade in excluded tumors, or Wnt inhibitors with CCFR inhibitors in ignored tumors. And so really this reflects the renewed efforts to delineate immune contexture through disease progression to get a handle on biomarkers. So this is a very active area, and increasingly we're also seeing efforts to move immunotherapies to earlier stage disease where they may have greater efficacy. So there's many ongoing efforts in this area. And I'll just highlight recent work from Carter and colleagues who sought to investigate the immune landscape of early stage PD-L1 positive breast cancers, looking at both RNA-based signatures as well as spatially defined protein signatures, with the goal of ultimately informing combination immune therapy strategies. Now, we know that TILs have been associated with higher rates of pathologic complete response to neoadjuvant therapy in TNBC, but this is not all consistently predictive. And so in this study, Yam and colleagues sought to look beyond TILs to more fully characterize immune response based on immune phenotypes and the association with neoadjuvant therapy. And then in related work, the I-SPY investigators sought to identify biomarkers that could discriminate between patients who would benefit from the addition of immunotherapy to neoadjuvant chemotherapy. And they focused in on the MHC class II expression profiles as a potential predictor. And so really there's a lot of activity in this space. I think ultimately any such biomarkers will need to be demonstrated to be technically reliable for diagnostic use, and stay tuned for further developments here. So we've touched on metastasis throughout, but just to recap on some of the key considerations around this, I want to just point out that, of course, there's increasing efforts to now atlas tumors through therapy and metastasis. We have a whole new set of tools to begin to profile not only single cells, but to profile tumor tissue in its native microenvironment, both at the transcriptomic and proteomic level. And so this should provide new insights into disease progression. I'll say that there was a lot of discussion around this at the Spotlight Session 6, focused on genomic and genetic analyses of metastases, including data emerging from the Metastatic Breast Cancer Project, but also real-world data and insights that can be afforded from such data sets. So briefly, I'll also touch on the European Aurora project, which sought to profile at the genomic and transcriptomic level paired primaries and distant metastases. Now, in this study, based on the expression data, they find evidence for subtype switching in 37% of cases, and this is predominantly amongst luminal-like tumors. They further evaluate ESMO scale for clinical actionability, or SCAT, and they find that roughly 50% of patients harbor SCAT tier 1 or 2 alterations, 36% if HER2 is excluded. And this is obviously important. I'll just call out important discussion this week from Fabrice André and colleagues in the Sapphire trials, which actually evaluated the clinical utility of genomic profiling in patients with metastatic disease in a randomized trial. And critically, they found that patients who received drug matched to their alteration had significantly better progression-free survival than those who received maintenance chemotherapy if they had a tier 1 or tier 2 alteration, whereas no such benefit was observed for patients with tier 3 or tier 4. And so really, this is one of the first studies to highlight the value of clinical decision support in treatment decision-making in the metastatic setting, with obvious implications for how we go forward. So just to close, really, there's a number of opportunities for further advances in understanding metastatic evolution and immune evasion, as well as therapeutic adaptation and resistance. And this will be supported by longitudinal profiling efforts, clearly also by trial-embedded molecular correlates and real-world data. We also need to better improve our understanding of resistance and paths to overcome this, both to endocrine as well as CDK4/6 inhibitors, but a whole host of other targeted therapies. Importantly, this will be enabled by improved biomarker stratification. And we also need to better position immunotherapies earlier in disease and to understand opportunities for combining approaches. Now, we didn't touch on this much, but understanding and ultimately therapeutically exploiting tumor dormancy is going to be critical, especially in late relapsing ER-positive, HER2-negative disease. And this also speaks really to the value of bringing forward ctDNA-based approaches to monitor treatment response as well as minimal residual disease. And really, we need to contemplate where elevated surveillance is necessary in the post-neoadjuvant and post-surgical settings. So another exciting area is in the development of biomarkers to enable therapy de-escalation. This is, of course, most advanced in the HER2 positive setting, but really we hope to see these opportunities move forward for other subgroups of disease so that we can spare patients unnecessary toxicity. So really much exciting work in the field and yet much more to be done. And I'll just close and thank you all for your attention, and I look forward to seeing you next year.